Okay, so you should have watched that video about Mendel and his pea plants. So again, dealing with our genetics, we have that trait. That's going to be that physical characteristics. It could be like height or color. Heredity is passing those traits from parent to offspring. And alleles are different versions of those traits. And so before we talked about homologous chromosomes that are, we get one from mom and one from dad. So they're going to have the same genes. Maybe allele here we have for flower color, but we can have different versions or alleles of those genes. So we can have white or purple for our different colors. Eyes would be another example. We have blue eyes and brown eyes. Those are two different alleles for the same gene dealing with our eye color. Okay, and so that's shown here. Again, we have two different alleles, and they're going to have differences in their DNA sequence, and that is going to affect the enzyme or the protein that's produced from that, and affect if it's functioning or not is usually how we're getting these different phenotypes associated with the different alleles. So we have something called our dominant allele. It's going to be the version that is expressed or where it is shown. So in this case, if we have two versions of DNA and one makes a protein that is functioning and one makes a protein that is not, even though we only have one copy, we still have a functioning protein and can make a functioning enzyme. So it's still going to be dominant, it's still going to be expressed. Other phenotypes would not that would not be expressed would be our recessive allele, which would be here. And so it's not expressed, and we would only see it present if both of the alleles present on the individual are the recessive allele. Okay, we can use some words to describe the inheritance of these alleles. Homozygous meaning the same variation is on both of those chromosomes. Heterozygous, they're different on each of the chromosomes. So, and we use capital and lowercase letters to denote this. Homozygous, here we can have homozygous dominant, homozygous recessive, or heterozygous where we have a mixture. The genotype is going to be written as this. So our capital letters, lowercase letters, these are our genotypes. So we have 3 different genotypes here that are possible. Here are some other examples showing how even though on one we have capital, lowercase, lowercase, capital, we can still write, we typically write our heterozygous genotype as big B, little b, even though it's really the same idea as this little b, big B. So the phenotype then is how that is physically represented, the green seeds or yellow seeds or whatever physical characteristic we observe. Okay, another important thing that is related to meiosis and our inheritance is crossing over and independent assortment. So these lead to 2 different laws of genetics, the law of segregation, which is the idea that our different chromosomes, those cells or those chromosomes are going to segregate into different cells, so they separate when they end up in different gametes, meaning that we only get one of these options from each of the parents. So we get one single trait. Each allele is going to separate into a separate gamete. So you see these are actually all showing homozygous individuals, but we only get one copy from each parent. And this is established in that metaphase 1 when we have that separation after crossing over happens of the 2 homologous chromosomes. The law of independent assortment is also dealing with meiosis, and this is the idea that the different traits or different chromosomes will assort independently. So genes on separate chromosomes assort into gametes independently. So here we have 2 different chromosomes. We have ones that are blue and ones that are red showing here. And in this first example, it just happened that both blue ones went into one cell and both red ones went into one cell. But it doesn't have to be that way. It could be that one blue and one red go into one and one red and one blue go into the other one. So it's just saying that they arrange themselves independently of each other, and this allows for lots of different combinations of gametes to be produced. So these are all important things to take into consideration and that contribute to genetics and how that those chromosomes are inherited. And this is again in metaphase 1 when they're separated out. Okay, so we're going to practice with an example. So a man is heterozygous for detached earlobes, which is dominant, capital E. He has a child with a woman who has attached earlobes, which would therefore be recessive. What is the chance that their child has attached earlobes? So this is where and there's a picture of detached and attached earlobes. So which phenotype is dominant? And so we already mentioned that the capital letter is going to be the one that is dominant, as well as the one that will be more likely to be expressed. What is the genotype for the man and for the woman? So the genotype for the man is heterozygous because it tells us, so capital E, lowercase e, and the woman has attached, so we know that that is recessive and it will be lowercase. What genotypes lead to detached earlobes and which to attached? And so we have two different options for detached. So we have homozygous dominant and heterozygous, and homozygous recessive for attached. We can also do Punnett squares, which you probably remember from regular biology, where we just take the genotypes of the individual and we put them over and to the side, and then we can fill in. So we have capital E, and lowercase. So in this case, 2 out of the 4 can be heterozygous, and 2 out of the 4 will be homozygous recessive. Okay, so we can look at the phenotype or the genotype. It depends on what they're asking for. So if we look at the phenotype, we can write that as 2 will be attached and 2 will be detached. So we can write that as a 1-to-1 ratio because we can just divide these numbers and write that as a ratio as 1-to-1. The genotype in this case is actually also the same, 2 and 2, and we only have 2 options of genotype. Okay, so these are some different Punnett squares that are always going to give you a phenotypic ratio of 100%. So this is important because normally when we're doing things with genetics, we are not actually measuring the genotype, but we can measure the phenotype. So a lot of times we'll do a cross, and then if 100% of the offspring are something, we can infer some information about the parents' genotypes. So it is important to know what are the options for getting 100% of the offspring to be a particular phenotype. So if they're homozygous dominant, or if we have one homozygous dominant individual and one heterozygous dominant individual, all of these would give 100%. 100% of the offspring would have the dominant phenotype. So these are ones that it's important to remember because we're going to need these later. There's also ratios we can have where we have 2 heterozygous individuals. In this case, we're going to have a 75% of the population that it's produced, or the offspring, will be dominant, and 25 is recessive. So a 3-to-1 ratio, 3/4 of the group and 1/4 will be different. So this is also a percentage that we will need to know. If we see 75-25, we should think it's going to be 2 heterozygous individuals. Okay, we can also get instances where we have 50-50 split. This is where we're going to have one individual who is homozygous recessive and one individual who is heterozygous. In that case, we have half of the individuals being heterozygous and half being homozygous recessive. So we'll need to know this as well. And then if we end up with all of the individuals being recessive, that means both parents also had to be recessive. So another example that we need to know. Okay, so doing these is kind of what we call a test cross. So we breed a dominant phenotype with an unknown genotype. And we will do this to determine the identity of an unknown. So how this would work. So if we know we have purple plants and white plants, and we do a test cross. In this case, our dominant purple plants are homozygous, and our recessive purple plant or blue plant, or what color is that? White plants have to be recessive and homozygous. If we get 100% of the population being purple, we know that the parent that was purple was homozygous dominant. If, on the other hand, we get 50-50, we know that Obviously, the white one was homozygous recessive, but that means that the other parent that was purple was not homozygous, but heterozygous on the other side. So a test cross, we are doing this to tell what is the actual genotype of this purple individual, because we can't tell just by looking at it. So this is getting at our idea of true breeding and purebred. We use these terms to talk about homozygous individuals. So they're going to always pass down the exact same genotype. They're homozygous. They can be dominant or recessive. So in this case, the white is considered a purebred because it's always passing down that white lowercase recessive allele. But the purple, depending on if it's heterozygous or not, might not be true breeding or purebred, but we can see when we get 100% of the offspring, it is a purebred. Okay, so we usually refer to this as the parent cross. So we're mating two true breeding parents to produce what proportion of offspring. We just did this one earlier, is we would get 100% of the offspring being heterozygous. And we refer to this sometimes as our F1 generation. So now we know that these are all heterozygous. And that's important because these new offsprings are purple, but we also know we have some seeds that are heterozygous. We have some seeds that we know are homozygous, and we can use those to do different tests. Okay, so if two individuals of the F1 are crossed, so the F1 would be this population here, we could then cross two heterozygous individuals and look at the ratio that we get as well. Sometimes they'll even cross an F1 with one of the parents, so one of those homozygous individuals. It's just ways to tell what type of inheritance we're getting. And this is also referred to sometimes as our F2 generation. Okay, there are multiple ways a genotype can be dominant. It could be, like we mentioned before, that maybe the recessive version of the gene is non-functioning. So the dominant is a functioning protein, and the recessive is a non-functioning protein. Okay, an example of this is albinism. So this is a genetic defect, and it's where the enzyme helps the body to change the amino acid tyrosine into pigment. So it's dealing with an enzyme. So inhibitor alleles prevent recessive alleles from expressing themselves. So those recessive alleles are not able to express fully, so they're like broken. Another example is the white feathers on a white leghorn chicken. Has an extra exon included in its mRNA, which prevents the expression of colored feathers. So it could have something, again, dealing with the genes and affecting what is expressed. An allele is dominant because it does something new is another reason we can have a dominant phenotype. So maybe it changes the function of the protein and it is able to gain a function. So an example of this is in dwarfism. So it's a broken version of a specific gene, FGFR3 gene. Normally its function is to stop bones from growing, but a mutated gene actually sends the signal too early. So it's still functioning, it just does a slightly different job. So it's still going to be dominant because even if we just have one copy of this enzyme, we're still going to have early signals. Okay, but what if you wanted to find the chances of offspring having two traits and not just one? And this is going to come up a lot in our AP Bio examples. So we want to find the ratio for maybe two different things at the same time. So we can do our Punnett squares as well. These are called dihybrid crosses, where we have two different traits that we are looking at the same time. So maybe we want to know the probability of having black hair and long hair for cats or something. Okay, so there's a couple different ways I'm going to show you. This first one is probably the one you're most familiar with, but it's actually, I'm going to go through it kind of quickly because I think there is a faster way that you should be able to know and utilize in AP Bio, especially since we have that timed test at the end of the year. So usually we think about using FOIL, where you are using first, outside, inside, last to get all of the possible combinations. So if we have hair color being capital F or lowercase f, and hair length being capital G, lowercase g, each each organism will get a copy of the F gene, or 2 copies of F and 2 copies of gene. So we can look at all the different combinations that we can get. If the individual is heterozygous, that means the alleles they could pass on would be first, so both dominant, outside, dominant F, recessive G, inside, lowercase f, dominant G, and recessive F and recessive G for our last. So this is just showing FOIL so that we're able to get all the possible options. It's basically just rearranging all of these so that we get every possible combination. And so what we would do then is we would use these to make our Punnett square. Okay, so if we have 2 individuals here with 2 different genes, we would get the gametes for one, get the gametes for the second, put it on a big 4x4 Punnett square, and then fill them all in just like we normally do. And you can notice that this is going to take quite a long time. And then once we do, we have to figure out, well, so which ones would be for long black hair? And it's actually all of these. And what if we miss one? It's gonna make it really difficult. So it makes our ratios really large and sometimes difficult to work with. But you can, if you are confused, always fall back on this method. It is perfectly fine to use. Okay, here's another example where we have different ones. So it's possible that we only have 2 different options if we have a homozygous individual. So because of that, we can shorten our Punnett square so it's not quite as long and just put those 2 on this side. So if that's the case, it goes a little bit faster, but we can still use that to determine the number of individuals who will have each one. Okay, again, for monohybrid crosses, this is pretty easy. We're just using one characteristic. But dihybrid crosses, like we were just looking at, have 2 characteristics like flower color or seed shape and take much longer to look at. But we also have trihybrid crosses where we have 3 genes or even more than that. And putting together a whole table like this just would take way too long. So instead, I'm going to show you what is the faster way to get these probabilities and proportions from that as well. And that is to use rules of probability. So we have to think about and or. So if it's asking for something dealing with and, we want to see individuals who have both black and long hair. We're going to multiply our 2 probabilities. If the thing is asking for something or, which won't happen as much, but if we're asking, okay, we want to know the individuals who have black hair for cats, but also, or if it has long hair. So that won't happen as much, but if that comes up, we will be adding them. Okay, so these rules of probability are also located on your equation sheet. The big thing to know is, and we multiply. Okay, so if you forget, they're there. So we're going to look at that same example, but now we're looking again at doing it a slightly different way, and it'll be faster. And we only do small Punnett squares. So in this, we're going to first just do a Punnett square with our F's. So notice that we have 2 heterozygous individuals. We have that here, and we can very quickly see 2 heterozygous. We're going to get a 3-to-1 ratio, 3/4 to 1/4. Okay, now let's do the same with G, with the color. So you notice also heterozygous, we also get a 3-to-1 ratio. Okay, so now we want to know long and black hair. So we look at what long hair is, 3/4. We look at black hair, also 3/4. We want both long and black, so we're going to multiply. So 3/4 times 3/4, if we do that, we get 9/16. So it's a much faster way of getting the exact same answer. Long and white, we have, again, 3/4. White this time is 1/4. So 3/4 times 1/4, we have 3/16. Short and black, we'd have 1/4 times 3/4. So also 3/16. And then that short white hair would be 1/4 times 1/4 or 1/16. And so how we write this as a ratio is it is a 9-to-3 to 3-to-1 ratio. And this is another ratio that's going to come up very often, and that is when we have 2 heterozygous individuals who are heterozygous for both genes in a dihybrid cross. It's going to be this 9-3-3-1 ratio. So remembering this 9-3-3-1 ratio and that it means heterozygous all around will be very helpful when we're trying to figure out some of these problems. But that's all we're going to do today. So we'll have more practice with doing these Punnett squares and having multiple dihybrid crosses. So, bye guys.